Welcome to my channel. I am Vishnu, second MSc Microbiology. Today I will take for amino acids roadmap. What are amino acids? Structure of amino acids, classifications of amino acids, optical properties of amino acids, acid-base properties, buffer characteristics, standard and non-standard amino acids, difference between exentyl and non-exentyl amino acid, and last of all discussions. What are amino acids? They are molecules containing an amino group, a carboxylic acid group, and side chains that is specific to each amino acids. The key elements of amino acid: carbon, hydrogen, oxygen, and nitrogen. Amino acids are basic structural building units of protein and other biomolecules. They are also utilized as an energy source. Structures of amino acids. Each amino acid except for proline, a carboxylic group, an amino group, side chain R group bonded to the alpha carbon atom. These carboxylic and amino groups are combined in a peptide linkage. This is a structures for amino acids. Classifications of amino acids. They classify according to the side chain. Amino acid with non-polar side chain, aromatic R groups, amino acid with uncharged polar side chains, positively charged basic for R groups, amino acid with acidic side chain. Classifications of amino acids for continue. Amino acids are classified as non-polar, hydrophilic, polar, hydrophilic, and acidic hydrophilic, basic hydrophilic. Non-polar side chains. The side chains cluster in the interior of the protein due to hydrophobicity. The side chains of proline and its alpha amino group form a ring structure. Proline gives the fiber structures of collagen and interprets the alpha helix found in globular proteins. For example, in nonpolar chain side chains, alanine, cytosine, glycine, isoleucine, lysine, methionine, phyllosine, proline, tryptophan, triosine, and valine. Aromatic R groups. Their aromatic side chains are nonpolar so that participate in hydrophobic interactions. Triosine is important in some enzymes. Most proteins absorb light at a wavelength of 218 nanometer due to aromatic groups, a property exploited by researchers in the characterizations of proteins. For example, for aromatic groups, phenylalanine, triosine, and tryptophan. Uncharged polar side chains. More hydrophilic because they form hydrogen bonds with water. Includes serine, threosine, cytosine, asparagine, and glutamine. Cytosine contains a sulfutryl group, an important component of active sites of many enzymes. Two cytosines can become oxidized to form dimethyl cytosine, which contains a covalent cross-link called a disfold bond. For example, for uncharged polar chains, cytosines, threosines, triosines, asparagine, and glutamine. Basic R groups. The R groups have significant positive charge. Lysines as a second positive amino acid group, argines as a positively charged gunitio group, acetine as a positive imidazole group facilitates the enzymes catalyzed reactions by serving a potent donor and acceptor. For example, for basic R groups, arginine, lysine, and histidine. Acidic side chains. Aspartic and glutamic acid amino acids are potent donors. At natural pH, the side chains of these amino acids fully ionized. They have a negatively charged carboxylic group. For example, for acidic chain, aspartic and glutamic acid, glutamate and amino acids. Optical properties of amino acids. The alpha carbon is attached to 4 different chemical groups called optically active carbon atom. Glycine is the expectations amino acids exist in 2 forms, D and L, that are a mirror image of each other. All amino acids found in proteins are of the L configurations. Acidic and basic properties of amino acids. Amino acids aqueous solutions contains weakly acidic alpha carboxylic groups and weakly basic alpha amino groups. Each of the acidic and basic amino acids contains an ionized groups in its side chain. Thus both free and some of the combined amino acids in peptide linkage can act as the buffers. The concentrations of weak acids and its conjugate base described by the entrance as bleach regulations. Buffer characteristics of amino acids. Amino acids have the capability of reacting with both acid and base. They naturally act as a buffers. The carboxylate in ion group act as a base and the amino group act as a acid. Example of buffer characteristics in amino acids: aspartic acid, one of 2 acetic amino acids. Aspartic acid and glutamic acid play important role for general acetic acids in enzymes active content centers, as well as in maintaining the solubility and ionic characteristics of proteins. Standard amino acids. Amino acids join together to form short polymer chains called peptide or longer chains called either polypeptide proteins. These polymers are linear and unbranched, with each amino acid within the chain attached to 2 neighboring amino acids. 22 amino acids are naturally incorrupted in the polypeptide and are called proteinogenic or natural amino acids. Of these, 20 are encoded by universal genetic code. The remaining 2 are incorporated. Into proteins by unique systematic mechanisms. Non-standard amino acids. Acid from the 22 non-standard amino acids. Acid from the 22 standard of amino acids. There are many other amino acids that are called non-proteogenic or non-standard. They are either not found in proteins, for example carnitine, or not produced directly. Non-standard amino acids that are found in proteins are formed by post-translation modifications, which is modifications after translation during protein synthesis. These modifications are often essential for the functions or regulations of a protein. For example, for non-standard amino acids, carboxylations of glutamate allows for better binding of calcium cations. In human nutrition, when take up amino acids into human body from the diet, the 22 standard amino acids either are used to synthesize proteins and other biomolecules, or oxidized to area carbon dioxide source of energy. The oxidation pathway starts with the removal of amino acid group by a transamine. The amino acid is fed into the urea cycle. The other product of transaminations is a ketoacidate that entire the citric acid cycle. Glucogenic amino acids can also be converted into glucose. Difference between essential and non-essential amino acids. There are 20 different amino acids that make up all proteins in human body. These amino acids required in tissues, red blood cells, enzymes and other substances. 9 to 12 amino acids can be manufactured of the body. Non-essential amino acids are not obtained from the thyroid. The remaining 8 to 11 essential amino acids must be obtained from the thyroid. Optimal obtained from nutrition synthesis of the body to for essential and non-essential: lysine, isoleucine, valine, histidine, lysine, methionine, phenylalanine, thiosin, tryptophan. This all the for essential, and non-essential for allergen, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine, tyrosine. This all for non-essential. Thank you.